Chemistry of DNA fingerprinting by Sierra Gainey. DNA fingerprinting is a test to identify and evaluate the genetic information called DNA in a person's cells. DNA stands for deoxyribonucleic acid. DNA was first identified and isolated by Frederick Miescher in 1871. DNA fingerprinting was invented in September of 1984 by genetic scientist Dr. Alec Jeffreys. Examples of DNA fingerprints: blood, hair, skin, saliva, urine, semen, etc. You can find DNA from many different ways from a person as long as you have some of their cells. What is DNA fingerprinting used for? DNA fingerprinting is used for many different reasons. For example, to find out family relationships, who the parents are of a child, find out if you're related to another, just to know any relationship you have with anybody, or if you do. Crime scenes, identifying bodies, identifying suspects or a person who committed the crime. When they commit crimes, they usually leave DNA somewhere, so the detectives collect that and give it to the scientists, and they identify who it is. Also, to identify organisms that cause disease. You can find out if you have a disease that might run through your family, that a family member might have passed down to you, or that you just have. Process of DNA fingerprinting. First, you have to have or find DNA cells. Then you have to isolate the DNA and amplify it with PCR. Then treat the DNA with restriction enzymes. Scientists then observe the different size fragments that separate DNA based on fragment size, called gel electrophoresis. Then fragments of DNA from restricted enzyme cleavage are separated from each other when they migrate through a support called agarose gel. Then the size-based separation of molecules of DNA separate based on size when an electric current is applied to the agarose gel, electrophoresis gel. Then, using nylon membrane, the separated DNA fragments are then drawn out of the gel. The nylon membrane is treated with chemicals that break down the hydrogen bonds in DNA and separate the strands. The single-strand DNA is cross-linked to a nylon membrane by heat or UV light. Then the nylon membrane with a radioactive probe of single-stranded DNA complementary to VNTRs, variable number tandem repeats. Now I have a video kind of showing what the process of DNA fingerprinting.
Enzyme is added to the samples to be compared. The enzyme cuts the DNA into smaller fragments. The DNA fragments are placed on an electrophoretic gel. When an electric current is applied to the gel, the DNA fragments migrate through the gel toward the positively charged pole. The smaller the fragment, the faster it will migrate. After a set time, researchers identify fragments of different lengths by staining the gel or by specifically highlighting fragments that contain tandem repeats.
And back to the slideshow. The chemistry behind DNA fingerprinting. DNA molecules are made up of hydrogen, carbon, nitrogen, oxygen, and phosphorus. With those 5 elements, it produces guanine, adenine, thymine, cytosine, phosphate, and deoxyribose. DNA is like a spiral 8 looking shape. DNA is all throughout your cells, so you have these molecules everywhere in your body. DNA is always made up of these elements, but what makes everybody different is the layout and where each element is. And as you can see, the DNA polymer shows where the hydrogen, carbon, nitrogen, oxygen, and phosphorus are, and where the guanine, adenine, thymine, cytosine are. Question: what chemicals are involved with DNA fingerprinting? Chemicals are used when trying to separate DNA from the rest of the cellular components. First part is the scientists use phenylchloroform to remove the proteins from the DNA. The phenyl and chloroform denatures and dissolves the proteins. Then they add salt, which interrupts the hydrogen bonds between the water and the DNA molecules. The DNA is then precipitated from the protein in a subsequent step with isopropanol or ethanol. The ethanol induces a structural change in DNA molecules that causes them to aggregate and precipitate out of the solution. Finally, the DNA is pelleted by spinning with a centrifuge and supernatant removed. Measurements and structure of DNA. DNA is measured in nanometers and microliters. The structure of DNA is a double helix. It's formed by base pairs attached to the sugar phosphate backbone. DNA is basically just made up of 4 nucleotides. Each nucleotide is made up of sugar, deoxyribose. The deoxyribose holds the phosphate molecule, and on the other side, it holds the nitrogenous base all together. DNA is made up of nitrogen bases called purines and pyrimidines. The purines are adenine and guanine. The pyrimidines are cytosine and thymine. The nitrogenous bases are then held together by hydrogen bonds. DNA is located in the nucleus of each cell that you have. And in the very left bottom picture, you can see how you get to the DNA. And on the very right, it shows where the hydrogen bonds are in the middle and how they hold thymine and adenine together and guanine and cytosine together. And it shows the deoxyribose, phosphate, all together. And this is more examples of how the structure is all together and how the deoxyribose sugar molecule holds the sugar phosphate backbone and the nitrogenous basis all together to form one DNA molecule. And on the right it shows what a purine and pyrimidine does, how they are shaped. Chemical formulas in DNA. Adenine is carbon 5, hydrogen 5, nitrogen 5. Guanine is carbon 5, hydrogen 5, nitrogen 5, oxygen. Cytosine is carbon 4, hydrogen 5, nitrogen 3, oxygen. Thymine is carbon 5, hydrogen 5, nitrogen 2, oxygen 2. Phosphate is phosphorus, oxygen 4. And then 2-deoxyribose is carbon 5, hydrogen 9, oxygen 4. Fun facts about DNA fingerprinting. DNA fingerprint scanners are used in phones now to tell whose phone it is. For example, in iPhone 6s and 5s, they have on the home button a DNA fingerprint scanner. That way, that person don't have to unlock it if it's their phone because it shows their fingerprint and it's the same fingerprint every time. And Yeah, everybody's DNA is different, so if someone else tries to hack into it, they can't do it because their DNA don't match their DNA. Then everybody's DNA is different, but twins can have the same DNA. DNA can be contaminated. Only one sample is needed to get a person's DNA. Once they get the one sample, that's all they need to get DNA. It's just a few cells, if not one. A person's traits can be found in their DNA fingerprinting because of their DNA sequences. Conclusion: DNA fingerprinting is way more complex than everybody might think it is, but it has made a tremendous change in this world. By solving crimes, finding family relationships, and finding diseases on many people's genes, it's impacted how we live and has helped us for the better. DNA has a lot of science to it. If it's biology, forensics, or chemistry, science has helped us for the better. Yeah, that's DNA fingerprinting.